Okay, thank you. So as people are getting their snacks, let me introduce sort of this next segment. And so we're going to have for this third session, going to be on biological factors in mental health disorders. And we'll be having three speakers today, Dara Rubinski, Michael Hallquist, and Christina Neely. And I'm going to start off with Dara Rubinski, who got her PhD from SUNY Buffalo and then did her fellowship at Hershey, where she is currently assistant professor in the Department of Psychiatry. And she is also a Birch Scholar, which stands for the Building Interdisciplinary Research Careers in Women's Health. And her interests focus on examining the long-term interpersonal and affective outcomes of ADHD in girls and developing treatments to address these. And so with that, I'd like to ask Dara to come up. And I'm going to ask that you give her a round of applause as she comes up, because I find that awkward walking silence. So let's give her a hand.
Okay. So I'm going to talk now about some of my research focusing on the risk for depression in girls with ADHD, and start to hint at some steps that I'm taking towards dipping my feet into the translational research. So to start off with a little bit of brief background on ADHD, typically a disorder that we see in childhood, primarily in boys, 2 to 9 times more frequently in boys than girls. We're all probably pretty familiar with the characteristics of ADHD. Really the only gender difference that typically emerges is that boys oftentimes present with the hyperactive, impulsive symptoms, whereas girls may be more likely to present with the inattentive symptoms. So most of what we know about ADHD, and there's a lot we know about ADHD, has really been focused on school-age boys. But we know that there's a substantial proportion of children and girls in the United States that have a diagnosis of ADHD, and there are really very few gender differences that emerge in symptom severity, in impairments, in treatment response by gender. One of the interesting trends that we see, though, is that girls oftentimes reach clinical attention later on, beyond childhood, into adolescence, in adulthood, and often in times where there's also co-occurring issues, whether it's depression or anxiety, that often co-occur with ADHD at that time. To talk a little bit more about ADHD and depression in girls, we know that in girls, regardless of ADHD status, girls are at least 2 times more likely than boys to develop depressive symptoms and clinically meaningful depressive symptoms. But when you look at comparisons between girls with and without ADHD, girls with ADHD are at risk for a range of different depressive-related outcomes compared to their non-ADHD girl counterparts. So things like earlier onset of depression, more frequent episodes, more severe episodes, episodes that are associated with more inpatient hospitalizations, non-suicidal self-injury, suicide attempts. But the one thing that we don't know is whether this outcome is specific to girls with ADHD versus boys. We know that in meta-analyses that have come out recently, anywhere between 20 to 50% of individuals with the diagnosis of ADHD will meet diagnosis for a major depressive episode across the course of their life. And we really don't have any samples or studies that to date have examined whether this risk is higher in girls with ADHD compared to boys with ADHD, except there's one that's come out about 7 years ago now that included 18 girls with ADHD compared to about 100 boys with ADHD, and also included comparison children, girls and boys without ADHD. And what we see in this study is that, yes, in fact, in this sample of 8 that included 18 girls with ADHD, beginning around pre-adolescence. So these were children that, at the left side of the figure, were in age 4, age 5. So beginning around age 11, you see that the girls up top are more frequently being diagnosed with major depressive disorder compared to the boys with ADHD. And a similar trend emerges below when you look at suicide attempts as a longitudinal outcome of ADHD, that girls are more frequently engaging in suicide attempts or are more likely to have reported one a little bit later on, so not pre-adolescence, but early adolescence. we see the gender distribution change. Okay, so this really interesting study with only 18 girls with ADHD, we really need to do more work here and really figure out what's going on and figure out the long-term outcomes of girls with ADHD. You need to incorporate more girls with ADHD, examine the risk for depression early on, so not at age 18 reflecting back on when you first became depressed, and understanding at what time developmentally are these depressive symptoms emerging for girls with and without ADHD and for boys with and without ADHD. Are there gender-specific effects? So one really opportune study to take a look at this would be to look in the MTA study. So this is the multimodal treatment of ADHD study, the largest treatment trial of ADHD to date, that included 4 treatment arms. Children were randomly assigned, children with combined type ADHD within the age ranges of mean age about 8, and were randomly assigned to one of 4 treatment arms, either the community control, where they could receive treatment in the community as you would typically do, and a large portion of those children received stimulant medication. Also, there was a treatment arm for stimulant medication, another one for behavioral modification, which included parent management training, social skills through the summer treatment program, and also school consultation, and then a combined treatment arm. So this includes nearly 600 children with combined type ADHD. About 100 of them are girls with ADHD. And then this treatment trial lasted 14 months, and about 2 years later from that baseline, children without ADHD were also included into the sample so that we could follow these children over time, and that this data is available in the public domain through the NIMH website, and these children have data into adulthood. So this is what we thought would happen when we looked at self-reported depressive symptoms over time in the MTA study. We thought that we would clearly see that the girls with ADHD, that blue line, would report the highest level of depressive symptomology, and that this effect, that the girls would stand out from the other groups pretty early on in pre-adolescence. We have very frequent, multiple data waves so that we would be able to see a nice figure like this that showed that, yes, even though these girls have been ignored in the research or not very well studied, this is really important and we should be focusing our efforts here. But in fact, what we found was this. There's really a lack or no growth over time. The girls with ADHD, if you think of that bar around a 13 as being the clinical cutoff of the CDI, this is the first version of the Children's Depressive Inventory. If we look at the group means, not really all that close, not really all that compelling, and this was really shocking to us. Maybe you see a little bit of a trend that the girls with ADHD are a little bit higher, but the scores are still pretty low. So we thought a lot about these unexpected null findings and started to think about why this was the case. So certainly it's possible and it's clear that not all girls with ADHD are at risk for mood disorders. And so looking at the mean level, we're going to maybe mask those differences. So it's really important to identify individual factors or high-risk portions of the sample that may be at particular risk for depression. And another important thing that I'll get back to a little bit later is that so children were in the MTA if they had ADHD and were treated as part of the study for 14 months. But after that point, into pre-adolescence, adolescence, adulthood, a large portion, nearly 70% of the children were medicated for ADHD at some point in time. So this potentially could affect and maybe mitigate the risk for depression in this unique sample. Okay, so I'm going to talk about 2 different studies now that have been offshoots of this exploration into the MTA depression findings. And so one of the things we started to think about in terms of figuring out maybe there are high-risk groups in this sample was to look at early pubertal status. So the literature on early pubertal status being associated with risk for depression certainly exists, but it's also inconsistent, where some research finds that early puberty is in and of itself a risk factor for depression, especially in girls, but in other work that doesn't come out, and it's not. So other people have focused on the accentuation hypothesis, and goes by other names, but that it's early puberty in the context of early adversity that together exacerbate the risk for depression. And so some of this work has come from looking at early puberty and girls with conduct disorder, girls coming from homes where a parent is depressed. But those sorts of characteristics overlap with a portion of families where a child has ADHD, but ADHD specifically in early puberty hasn't been examined. So we thought that potentially, if we look at girls with ADHD and we look at pubertal status, we might see that early maturing girls with ADHD are at particular risk for depression compared to the other 3 groups in the MTA. So as I mentioned before, we have a pretty large sample, pretty unique to have 91 girls, about half of them at this point. This is the pre-adolescent period, so this is only a cross-sectional examination at this point. When the children are about 11 years old, we have reports from parents and teachers about ADHD symptoms. We have the children self-report their pubertal status, and a portion of those children, pubertal status was also verified by a study nurse, and then the children are reporting their depressive symptoms. So we have a total score, and then we have these other aspects of depressive symptomology from the CDI-1. So we looked at 3-way regressions, ADHD by gender, by pubertal status. And so for total depressive symptoms, we did find our expected effects. So if you look at that red or pink, however it's coming up line, these are the early maturing girls. And so on the right end of this figure is the girls with high ADHD symptoms, and they're reporting the highest levels of depressive symptoms here. If you look at the later maturing girls, no significant effect of ADHD. But if you look at that blue line, this is later maturing boys who seem to show a similar pattern to the early maturing girls with ADHD, but in a different direction. So that, well, not a different direction. The later maturing boys look very much like the early maturing girls. And if you look into the individual subscales of the CDI, none of them seem to drive this effect, except when you look at the anhedonia scale. Very similar pattern for early maturing girls with ADHD reporting the highest levels of depressive symptoms. And then when you look at the later maturing boys, that blue line, the boys with ADHD who are later maturers are the ones that are reporting higher levels of depressive symptoms compared to those non-ADHD later maturing boys. So from this, we found, and these are very brief, we could probably talk lots more about these findings, but that the expected ADHD by gender effect that I showed those non-compelling bar graphs before didn't emerge, but early puberty did help identify a subgroup of girls with ADHD that were at particular risk for depressive symptoms, and anhedonia seemed to be driving it. So when I talk about dipping my feet into the translational research pond, the finding that emerged specific to anhedonia starts to pique interest in looking at reward processing and underlying neural underpinnings that may be really at the core of this ADHD depression nexus. Another important factor that I alluded to before was that treatment history may impact the risk for depression, and many of these children were treated for ADHD. There's some work that shows that stimulant medication could affect growth, maybe puberty, but very little is known in this respect. Okay, so study 2, we are starting to examine the effect of treatment, ADHD treatment, so those evidence-based treatments for ADHD, behavioral treatment, medication, their combination, and community control, and how that might affect the course of depression over time. Is there a differential effect by gender? So now we only have the children with ADHD, not the controls, and are looking at linear mixed models where we're looking at type of treatment by gender, by time. So this is going from baseline around age 8 all the way up into 9 years post-baseline. Okay, so on the right you see the girls. This is a little messy. And on the right you see the boys. And so we do actually find a significant three-way interaction here. And when you piece it apart, what you find is that for the boys, any treatment that you picked was associated with pretty similar reduction in depressive symptoms throughout that first part of the graph. So where the boys are in the MTA treatment trial, they're improving. Their depressive symptoms are going down. And then slowly into pre-adolescence, adolescence, you generally see a little bit of an increase over time, which we would expect with the age range to some degree. Now for the girls, even though the intercepts are a bit different, particularly for the psychosocial group, the green line, and the medication group, the pink line, despite that, you do see where we're seeing the effect is that girls in the behavioral treatment are decreasing in their depressive symptoms more than they are in the medication group, in the MTA treatment part. And that in the post-treatment period, after girls are just doing as they would typically do, and some of them are receiving medication, some of them may have behavioral treatment, and so on, but it's not part of the study. We see that in the medication group, girls still kind of stay at this steady level. There's a little bit of a dip, versus in the behavioral group, the green line, girls continue to dip a little bit more, continue to reduce in depressive symptoms, and then slowly start to increase again as they reach adolescence. So this was intriguing. Certainly we ran a bunch of different models, controlling for things like number of days on stimulant medication and the whole entire length of data that we have, controlling for things like ODD, and the same model seems to persist. Certainly this needs replication, although that would be very time-consuming. But these are small sample sizes. These are at best at any time point, there's about 100 girls with ADHD, and then over time that diminishes to some degree. And the sample sizes, when you divide those 100 girls into 4 different groups, gets even smaller. But perhaps this starts to fit with other developmental literature showing that girls may be more sensitive to behavioral intervention because maybe they're more sensitive to interpersonal conflict. So an intervention like behavioral parent training or school consultation where the teacher is interacting and providing lots of praise and reinforcement is potentially differentially associated with reducing or mitigating risk for depression compared to a medication that isn't necessarily targeting interpersonal functioning or mood, things like that. Okay. But this is really something that needs more replication and that I'm busy working on now. Okay. So I'm not sure how I'm doing on time. Okay. So really to wrap up, I think I want to push for more research on girls with ADHD and understanding the risk for depression more generally. Future research really needs to incorporate both genders and understand, rather than at a broad group-based level, what are these subgroups of girls and children more generally who are at particular risk for depression. Certainly there are translational models and research that we can integrate into this work, which is where I'm hoping to go. And so to that, I want to talk a little bit about the Her ADHD study, which is an ongoing research study in Hershey, where we're looking at these types of variables and recruiting a sample of girls with ADHD in pre-adolescence and adolescence, assessing mood and interpersonal functioning to better understand mechanisms underlying ADHD and depression. And so in addition to, the MTA is a great study and it's a really unique study, but it's a lot of self-report. And so to build off of that, this incorporates self-report, observations, laboratory assessments, starting to get ERP and EEG data on these girls, and moving forward in that direction. Okay, so one thing about this sample is many of the girls who come in are completing a one-time assessment, so we get lots of data on them, assessing these types of variables. But half of the group has also completed a behavioral treatment. So we can also start to answer questions about how girls are responsive to treatment and work on extending that pretty interesting but small research from the MTA study suggesting that behavioral treatment is differentially better than medication treatment in reducing the risk for depression in girls with ADHD. Okay, so that's all I have. I just want to acknowledge some people, mentors, support for this research, and finish up.
While Michael gets mic'd up, let me introduce Michael Hallquist, who is a professor of psychology and happens to be my next door neighbor in the Moore building. Michael got his PhD from SUNY Binghamton, so another SUNY person, another SUNY system grad. Michael is most recently coming from the Western Psychiatric Institute in Pittsburgh, where he did his internship and postdoctoral work as well. He is interested in understanding the development of psychopathology of borderline personality disorder, especially how psychosocial and brain development is linked with emotion dysregulation and impulsivity. In particular, he uses computational models of decision-making with model-based analyses of functional MRI data. So without further ado, I'm going to introduce Michael Hallquist. Let's give him a hand.
All right, so thanks to Denny and to Emma for inviting me to talk. I was mentioning to a few people earlier, this is a little bit of a different talk than I usually give. There will be some data from various ongoing projects in the lab, but it will also be more of a conceptual piece. I don't usually push myself to think about translation, so I will clumsily go through my thoughts on that topic. But I think the underlying message that I hope to convey is that I think right now there are not enough bridges between brain and behavior. I will talk about one in particular, mathematical or computational bridges, that I think can help us extend translational efforts from brain-related research into more psychosocial intervention. But I'll kind of start out by talking a little bit about what borderline personality is. So the DSM defines the diagnosis mostly in terms of 4 core features, well, it's technically 9 symptoms. They center around impulsivity. This includes self-harm and suicidality. Emotion dysregulation, where in borderline personality you tend to see vacillation in affect that's quite rapid, often in response to interpersonal events, perceived rejection, arguments, etc. There's a pattern of interpersonal dysfunction where relationships tend to run hot and cold, or to have kind of a split representation of idealization and devaluation. And finally, a pattern of identity disturbance where self and other representations tend to be disturbed and the sense of self is not coherent or consolidated. It's important to mention that in the psychometric literature, BPD tends to fall in between internalizing and externalizing disorders. These are seen as kind of superordinate dimensions of psychopathology, and BPD has elements of both. So you see both depression in BPD, commonly it's 70%, as well as substance abuse problems, impulsive aggressive behaviors, and so on. So it affects about 1 to 2% of the general population. It's slightly more prevalent, it seems, in adolescents, although the jury's still out on that. I do work on development of borderline personality, but I'll mostly not talk about it today, with one exception. I'll be focusing more just on the kind of scientific vision of how to move forward in this research area. So despite only affecting about 1 to 2%, it's disproportionately represented, especially in more acute treatment settings. I actually had an RA in Pittsburgh who started with me after working in an inpatient setting, and she came into the lab thinking that everybody with borderline personality disorder would be kind of constantly trying to kill themselves or abusing polysubstance all the time, and that's just not the case. But nevertheless, if you see people with BPD in treatment settings, they tend to be quite severe, and they unfortunately tend to be frequent users of inpatient hospitalization. There's a very high risk for suicide completion in BPD, and so it's a high kind of priority from NIMH and just for us generally as a field to try and address suicide in borderline patients. And there is ample evidence to support the construct of BPD, for example, heritability data and twin data supporting the heritability of the phenotype, especially of affective instability or emotion dysregulation. So this is kind of my big figure that I'll come back to, and that, you know, like somebody said earlier, I'll probably die before I complete this vision, but it's kind of how I think about the steps along the translational bridge in my own work. And the idea essentially is that we can talk about the latent structure of psychopathology. What are the disorders? What are the constructs we try to measure? That's, of course, been a big area of interest with the DSM-5 task force, especially for personality disorders, where there was a lot of debate about how to think about these mental health conditions. But we can also talk about specific clinical syndromes, for example, borderline personality. And historically, those have been thought of as clinical entities. This is, you either have it or you don't. It's the kind of diagnostic perspective on that. In large part, that's given way more recently to dimensional conceptualizations of psychopathology. For example, trying to identify underlying psychological features. So two that I'll think about, especially in borderline personality, is impulsivity and emotion dysregulation, where those are expressed in borderline personality, but not necessarily unique to that clinical syndrome. And yet, we can go even further and think, well, how do we tap into that? If we really want to probe something under the hood of emotion dysregulation in borderline personality, what can we do? In our lab, we focus especially on trying to develop experimental paradigms that tap into specific features, specific psychological constructs. So for example, if we think that reward processing is affected in borderline personality, then perhaps we're going to give somebody a task where they have to make reward-related decisions and try and characterize behavioral differences in people with BPD compared to depression or healthy controls. But we can also go further than that and try to develop mathematical models that bridge from the brain level, from kind of neural computation, to what we might see as behavior on a task in the lab. And that's where I'll spend some time later in the talk, but many of these fall under the kind of rubric of reinforcement learning models, where we try to predict somebody's behavior on a trial-by-trial basis. So I'll try and work through some of these to the extent that I can say anything intelligent about them. But the first question that comes to my mind is, at what level do we intervene? And historically, this has been thought of in borderline personality as either intervening on the syndrome or perhaps on psychological features underlying it. It's worth noting that 3 psychosocial treatments are the best validated, most empirically supported for treating borderline personality. And that if you look at, for example, the NICE guidelines, the NHS in the UK releases guidelines on treating different mental health conditions, they specifically do not recommend any medication for the treatment of borderline personality. And indeed, treatment trials, RCTs using different drugs like SSRIs or antipsychotics, mood stabilizers, are very inconsistent in borderline personality, and the evidence is quite mixed. So what do these 3 treatments do? Well, do they treat the syndrome? Do they treat the underlying psychological features? What are the mechanisms of those treatments? I'm certainly not a treatment researcher, so I don't know that I'm going to answer that question, but I think it is worth thinking about what's in common across those interventions. So in order to have an effective treatment for borderline personality, these 3 treatments have some common elements. One is that they focus on the therapist-client relationship. So if indeed intense, stormy relationships are a hallmark of BPD, often the relationship with the therapist needs to itself be curative in some way. There needs to be action in the room, so to speak, that the relationship with the therapist evokes the kinds of dynamics that are problematic in the world and therefore can be worked through in the course of therapy. Therapists who treat BPD need to be willing to work intensively with patients. In DBT, dialectical behavior therapy, for example, there's a model in which the treatment is quite comprehensive, and therapists will give clients access to their cell phone number. They'll have check-ins. There'll be kind of frequent consultation in some cases. There's group, there's individual, so it's often a quite intensive approach. There are less intensive versions of that, but it's worth noting that it spans the spectrum. Therapists provide direct feedback to clients about their behaviors in session. It's often seen as being kind of brutally honest or irreverent in some cases. Therapists need to target therapy interfering behavior, showing up late, bringing up something really intense and problematic in the last 2 minutes of the session, such that you have to run over. Contingency management, so being clear about what will we do if you try to kill yourself, or what will we do if you show up to the session drunk. Improving the mentalization and metacognition of these patients is really important. And so all these treatments have these kinds of ingredients in common. If we go to the other end of that spectrum and say, okay, well, what about the neural systems? What's affected there? So this is like my poles of my kind of distribution of levels of analysis. There's increasing literature on decreased connectivity between the OFC, VMPFC, subgenual ACC, all, depending on the study, all of that, and the amygdala. So this kind of ventral prefrontal amygdala connectivity tends to be somewhat blunted in BPD. We know from animal studies that this is a key pathway in inhibiting emotional responses and perceived controllability of stressors, like Meyer and Watkins' work. There's also some evidence that how people integrate social signals, especially social feedback, may be disrupted, and people with BPD may not perceive ruptures in the interpersonal relationship, and this could be related to anterior insula. Volumetric studies have found gray matter concentration is increased in the amygdala and decreased in rostral and subgenual ACC, so more of this kind of frontolimbic story. Likewise, we tend to see amygdalar hyperreactivity to emotional cues in BPD across a number of studies. And then last, there's decreased volumes of the hippocampus and amygdala. People have talked about, like in stress or in trauma, this kind of glucocorticoid toxicity hypothesis. So if we kind of put all that together, it's sometimes been called the frontolimbic hypothesis of BPD, but we've even heard today other frontolimbic hypotheses, for example, in eating. Not enough prefrontal, too much limbic. This is a common refrain, and I think many people in the kind of field of cognitive neuroscience more broadly are struggling with this. What's specific about that? So in the case of BPD, you can find analogs in mood disorders, like Yvette Chaline's work, or in anxiety, where this kind of frontolimbic notion prevails. Likewise, we don't necessarily know what the clinical implications of these neural abnormalities are. So it's not clear how do we take some of those key ingredients from the therapies I mentioned and think about, well, why would the subgenual ACC be showing blunted connectivity to the amygdala? There's just those are miles apart in terms of levels of analysis. And experimental data to date don't really tell us about the cognitive processes that are under the hood. Much of the work has been focused on facial perception, emotion perception, these kind of basic ingredients. So sometimes I'll ask myself something, like, what individuals with BPD show greater activity in a reward-related region when they're thinking about aggressing against somebody. Does that mean that they're finding that rewarding? That's a kind of classic reverse inference in cognitive neuroscience. If we see striatal activity while you're thinking about hitting somebody, does that mean that you think hitting people is rewarding? I think most people would agree that that's a kind of troubled perspective on this. And yet that's in some ways where the field lies right now, at least in BPD imaging research. So my proposal is that we have to work on both ends of that and try to build toward the middle. We need more work on refining phenotypes so that we have a better sense of what is BPD clinically. How do we think about it? It's often been called a heterogeneous disorder. Does that mean we need to do something about that? It's up for a debate. And likewise, we need to think about the neural circuits that are disrupted in a more detailed way. So just as one example of this, in some work I did as a postdoc, we did some subtyping research on different features in BPD, trying to separate out potential subgroups that have different clinical expressions. The short version of this is that we found one group that was kind of a prototypical group that had average levels of things like antisocial personality, aggression, paranoid personality. These are all intended to be features that are not necessarily baked into the BPD construct, but it can instead differentiate between subtypes of borderline pathology. But, for example, we found a group that had poor identity consolidation, that had identity disturbance, and a relatively low level of anger. These were folks that were not the, I'm going to lash out at you and hit you, but were more self-injuring, more self-blaming, and actually had poor identity consolidation. There was an angry, paranoid, somewhat suspicious group that tended to see threats out in the world much more readily. And then the most severe group was a group that had both anger and aggression in a more externalizing fashion, suggesting that there's, consistent with the clinical theory, there's this kind of anger that may be focused on the self and seeing kind of suspicion out in the world. There's also anger that's externalized in some individuals. So how anger is expressed and kind of affects people with BPD may be different depending on the person. And likewise, where identity fits into this may also depend on the person. So one kind of example of what you might do to try and look at the phenotypes, clinically speaking. So if we kind of bring ourselves back to this wild figure, I think one thing to think about as well is what's on the other side of that spectrum. So in some work that's being led by my grad student, Nate Hall, we've also talked about that perhaps intrinsic connectivity, sorry, oh, did you say something? Oh, okay, I'm just hallucinating. Okay. So in some work on the other end of the spectrum, there's, I think, increasing evidence and promise for a focus on intrinsic brain connectivity as a measure that can be used across different diagnostic groups, different populations, that may help us to differentiate what are the neural systems that are most affected in a given clinical syndrome or in a given psychological phenotype. And this is preliminary work, but one thing that we've found is that it seems as if the integration of the default mode with other regions, particularly other networks, so control-related networks, somatomotor, visual networks, et cetera, that there seems to be an abnormal between-network integration with development in BPD. That in most individuals, the default mode tends to separate with age. It tends to show decreasing connectivity. For those who are not in this world, the default mode is thought to be kind of what your brain is doing when it's just resting in the scanner. But we know that people don't just sit there. Nobody is kind of free of cognition for any moment in time. So it tends to reflect more self-referential processing, social cognition, maybe even planning, like what you're going to get at the supermarket that day. But studying people in this kind of resting state, the default mode tends to be the regions that are activated during that time. And so we see that in those regions, there's this abnormal integration with other brain networks that are involved in control and so on. So from a translational perspective, even these steps are not enough. And I kind of am saying this to myself at the bottom here. You know, it's the classic quote from Churchill. It's no use saying we're doing our best. You've got to succeed in doing what is necessary. And so this is where I tend to stop in my thinking. It's like, well, translation's hard. I don't know how to help anybody. I'm just a psychologist, you know. So what are the obstacles to translational progress? I think from a kind of eliminative materialism perspective, clinical theories of BPD are rich. This identity diffusion, for example, it's a rich construct. It's at the core. Wow, sorry. That was an uncontrollable stressor. It's at the core of one of the psychodynamic theories of borderline personality. And yet it's largely specified in terms of other fairly abstract inferential concepts. It's not clear how you kind of get under the hood of identity diffusion in a Kernbergian sense. It doesn't make such theories wrong. It just means that their explanatory power is not clear, or maybe at a relative level. So in epistemology, there's this idea of criteria of truth. You know, how do we evaluate what's true and what's not true? That's not baked into logic. It has to be kind of axiomatic. It has to be something that we agree on up front. So if we were to debate this issue, we'd have to say, well, on what basis can we judge a theory to be true or not? So what I'd like to suggest is that to translate from bench to bedside, you really have to build tangible bridges among levels of analysis. And I can only do my little slice of things that I'm, interested in and good at, which is a kind of relatively narrow intersection of things. But I think in BPD research, my agenda, and I think an important agenda in terms of clinical psychology or psychopathology research, is to identify and study phenotypes that reduce across levels of analysis, kind of spanning brain to behavior to clinical phenotypes, that are generally conserved across species. And so even though I love identity diffusion, I'm not really sure how to develop like a mouse model of that construct. Whereas emotion dysregulation or kind of mood instability, I think there's more promise there. I'm happy to talk more about that later. I think those phenotypes need to be operationalized in terms of causes and conditions that can be experimentally manipulated or at least rigorously quantified, and that these have plausible links to neurobiological systems. That would be the kind of very Patricia Churchland-like perspective on this, that theories can be useful to the extent that they can be biologically specified. She takes a hard line on it. I'm not quite there yet. But I think it's important to think about that up front in terms of what constructs are going to help us understand borderline personality. So I'll say a little bit about other steps here. And I think the step that I'm working on a lot right now is these what are called formal generative models. And these tend to fall right between psychological features and experimental manipulations. So if we want to say what leads you to do something on that reward processing experiment where you're gambling, well, what's giving rise to your behavior? We could, of course, look at mean reaction times or the number of errors you make or perseverative problems, failures to reverse and contingencies, those kinds of classic ideas. But a formal generative model says, actually, what caused that behavior? Can we simulate behavior from an agent that acts like you? And so I'll give just one quick example of that. Although before I do that, I just want to kind of recommend to your attention this recent paper from John Krakauer. He gets into a number of things that might be falling under kind of David Marr's classic idea of levels of analysis in neuroscience, going from kind of implementation level to computation. But I think it's a really important commentary that goes against some of the shifts in recent neuroscience toward big data or especially kind of technology-driven research. So we now have these incredible tools like optogenetics or CRISPR that can help us manipulate things at the synapse level or at the single cell gene expression level. But I think we have to keep an eye on studying behavior and really thinking about what are the behavioral targets, how do these map onto phenotypes that we care about. And I think there has been a shift in his view, and I would agree with it, too much toward getting into mechanisms that are purely experimentally and causally specified without thinking about how they link back to these behaviors that we want to study psychologically. So let's move ahead to one example of a generative model, and that is the drift diffusion model. For those who don't know about this, this is a model developed about 30 years ago to really look at speeded reaction time type tasks, like a go, no-go task, where you have an opportunity to, you're usually pressing the space bar, like A, B, C, D, press the space bar. But if an X comes on the screen, you have to not press the space bar. So it's an inhibitory control task where you have a prepotent tendency to press the space bar, but you have to withhold that. The DDM is one example of a generative model. It says you have two kind of directions your evidence is accumulating in. The stop direction, I shouldn't respond, or the go direction. And when the letter comes on the screen, you accumulate evidence toward one of those bounds. If the X comes on the screen, hopefully you go toward that no-go side. But if the A comes on the screen, hopefully you go toward that press the go button side. So the DDM actually specifies how data are generated from that, and you can test hypotheses about that in clinical data as well. So my last kind of wrap up here is that you can talk about emotion perception in BPD as having largely focused on a tendency to see negative stuff where it's not there, or maybe to being biased towards seeing negative faces or negative emotions on people's faces. So if you show a person with borderline personality a neutral face, they'll tend to identify it as potentially threatening, angry. But we don't necessarily know what's under the hood of that. So as one kind of generative example of how this sort of model can be applied, some work that my student Allison has been working on, we have a kind of emotional Stroop task. And in that task, you can think about how people accumulate evidence according to what's called a drift rate. And that's, the drift rate is essentially how quickly you move toward one of those bounds. So it would be like coherence or the kind of informative status of the stimulus. And what we see is that, especially for negative emotional cues, when the fear is the emotional cue that you see on the screen, regardless of whether it's a congruent or incongruent stimulus in the Stroop sense, that people with BPD tend to accumulate evidence more quickly toward that bound. They're biased in the direction of seeing fear where it's not there, but it's really that accumulation process that seems to be effective in BPD. If you show them a happy face, on the other hand, they're slower to accumulate evidence toward that and identify that affect correctly compared to people who do not have borderline personality. So yeah, I will stop there, but I think just wanted to highlight this as a kind of example of this, how do we build translational bridges between brain and behavior? I think some of these formal mathematical models can help us get there. So thanks.
Our final speaker is Christina Neely, and let me get my notes. We have somebody who's not from a SUNY school, so that's a change, right? So Christina Neely got her PhD from the University of Western Ontario, and did her postdocs at University of Illinois Chicago and the University of Florida. She's currently an assistant professor of kinesiology, and her research focuses on understanding the central nervous system, how the central nervous system organizes the preparation, execution, and inhibition of skilled, purposeful actions, and in particular in the production and inhibition of precision grip force in healthy individuals and patients with attention deficit disorder. So please join me in welcoming Christina Neely.
Thanks for the nice introduction. Thank you to Denny and Emma for inviting me and hosting us all here today. I'm going to talk about something a little bit different. I'd like to try to convince you over the next 15 minutes or so that we should care about movement and that it can tell us something important about disorders. Not just movement disorders. Movement disorders, it seems very clear, right? We see someone who's having trouble walking or reaching for something and, oh my gosh, that's a problem. We need to work to fix that. And so in the last, I've been here about 4 years now and been privileged enough to work with great people like Cynthia and Corrali and other psychologists like Dara who have helped me kind of realize and put this in context in terms of neuropsychology. If we have a patient who has borderline personality disorder or schizophrenia, a movement disorder is the least of our concerns, right? We've got to work on thought disorders. We've got to work on suicidal ideation and other important problems. But it's possible that if we measure movement, we might be able to learn more about the disorder. And I'm going to try to convince you at the end that it's possible we might even be able to treat the disorder through some movement-based treatments. So here we go. So when you heard the word movement, you might have thought, oh, okay, she's going to talk about muscles or something, right? So here's muscles. This is our output system. And when you hear kinesiology or movement, you might think of that, right? Or weightlifting or training, and that's true. So the muscles do the work. But something has got to tell the muscles what to do. And I love this figure because it gives us a really good picture about all of the different regions that are participating in you reaching out for that cup of coffee and not spilling it, right? Applying the right amount of force, getting it to the right place, putting it back down. And so where I operate is up here at the top. I'm really interested in these descending systems, and I was lucky enough to do a postdoc where I was really immersed in the study of the basal ganglia, and that's where I started learning about, wow, this structure is really involved in movement, but it's involved in almost every mental health disorder population. Should we be looking at movement in this group? So I'm going to talk about tasks today that involve motor cortex, but are also really targeted to basal ganglia structures. How do I do this? Well, I use systems neuroscience techniques. I do a lot of behavioral work. I'm really grateful for the reference there to the Krakauer paper about how important behavior is, because that output of the skeletal muscles, we can track that back to basal ganglia or cerebellum or motor cortex. So I do some basic work, behavioral work, and I shouldn't call it basic, behavioral work that quantifies motor output. We can do measures of cognition, executive function, neuropsych type tests. We can look at brain structure and function. And since I arrived here, I've been working with Helen Kamens, who's helping me learn how to integrate human genetics. And so from my perspective, I'm trying to look at how the basal ganglia and the prefrontal cortex are working together to generate movement. And the clinical model that I've been looking at since arriving here is ADHD. So here's the task that I use. It's pretty simple. This guy up here is our in-house model that we use in the lab, and this guy on the bottom, this one here, is our fiber optic grip force transducer. So these are just some simple steel load cells. We place them in opposition, and participants are going to push with their index finger and thumb in response to stimuli on the screen. Also pretty simple. This guy on the bottom, when it turns green, you need to press. When it turns red, you need to rest. And we can have it move up and down to give instant online feedback. What's great about this being really simple and just being 2 bars is that we can translate it to the MRI environment. It can fit really nicely on that little screen, and we're lucky enough to have this fiber optic transducer that we can also use in the MRI environment. So this is basic one-dimensional force. We're not lifting, we're not looking at slip or anything else. We're just looking at how do you create one-dimensional force in response to visual feedback. And we can make about 1 million experiments out of this because we can change the gain of the feedback, we can magnify it, and then we're looking at things like detecting error and making corrections. We can take the feedback completely away, and now we're looking at memory systems, and we can change the timing and so on and so forth. So it's a really simple task that we can apply in different settings. So this is great, as I said, for the MRI environment, and I did an experiment looking at these 2 tasks, really simple comparison between static force and what I'll call dynamic force. And the only difference between these is this component of on and off, or just maintenance. Okay, so for 30 seconds, you're either going to do these 2-second pulses with 1 second of rest in between, or you're going to maintain force. And when you do a task like this, you get really nice, clear, what we call visual motor networks. And so what I'm showing you here is just one of the figures looking at areas that were greater in the static task relative to the dynamic task. And then on the flip side, regions here that were showed greater activation and a larger extent of activation in our dynamic task. And so as we expected, the static task is going to involve a lot of error regulation. You're trying to maintain. And it also involved a lot of areas in right dorsolateral prefrontal cortex. In contrast, this dynamic network seemed to involve a lot of left lateralized regions. So now we have this nice network. This was done in healthy young adults. And we can then take these tasks and apply them to clinical populations without having to immediately run to the scanner, right? Because we know what the networks are. We know what regions are involved. We can do this with patients with essential tremor, Parkinson's disease, lots of disorders. And what we find is that, yes, as you would expect in a movement disorders patient, the movement is disordered in lots of different ways depending on the group you're looking at. But we also have differences in brain activation. So in the case of essential tremor, what we find is that M1, which is our primary motor cortex, and V1, our visual cortex, are hyperactive. Whereas the lobules of the cerebellum, which is where we know a lot of the pathology of essential tremor is in the cerebellum, are hypoactive or underactive. And so we can get these distinct patterns then in these clinical populations after we've characterized their behavior in the lab. So let me give you an example of kind of how I was already primed to study cognition after my graduate work. I was trained by a cognitive psychologist and was really interested in reaching tasks that involved some type of cognitive control. And then I was able to work with Parkinson's patients and atypical Parkinson's patients during my postdoc. And I'm not going to get into extensive detail about these groups, but it would be really great if you could distinguish among these 3 when you walk into the clinic. Because the problem is they present the same. But if you have PSP, you have a very, very rapid decline. If you have Parkinson's disease, you've got 20 years to go, right? You don't necessarily die from PD. PSP, you have 5 or 6, and it's accompanied by dementia. And you need to make decisions, and this information would be really helpful at the onset. But clinically, we can't distinguish these 3 groups, okay? And so we started looking at behavior. We used our task. Here, the difference is they're always going to the same target, and here they're going to a different target. So you could call this predictable and unpredictable. And we did this in all 3 groups. I know there's a lot of information here, but if you just look at the pattern, what you see is that here, the healthy controls are different from everyone. That doesn't help us. You could tell me that from across the room, right? And we have that for all of these different force measures, until you get into things like force pulses. And so we looked at our standard measures, like how quickly do they get up to the target? How quick do they go down? That's a key symptomatic trait for Parkinson's, right? They're slow, they're bradykinetic. And all of our standard force stuff wasn't differentiating these groups, but as the lucky postdoc in the lab, I had to hand mark all this data. And so I knew the data very intimately, and I said, but these guys who have PSP, they just keep pressing and pressing and pressing. And so if we take that time, let me go back. I don't have a figure showing the entire run, but after this 30 seconds, we have 15 seconds of rest, and then we do it again. And we do this 4 or 5 times. And we use that 15 seconds of rest, we subtract that brain activity from our active period. That's how we do basic task-based fMRI. And I said, but they're going to continue to do the task. This just doesn't seem to work for us. So I really started to pay attention to what were these PSP patients doing when they were supposed to rest. And they just kept pressing and pressing and pressing. And I found this really interesting. I happened to be already interested in inhibitory control, but started to think this is a disorder that is characterized by dementia. We're saying they have movement problems, and we're attributing that to a movement disorder, but there's a cognitive component to especially all of these contrived tasks that we do in the lab setting. And if we're not accounting for that cognitive component, then we might be blaming something on the motor system that is incorrect, or vice versa. And so I, at one point, gave a talk and I said, I'm going to disentangle cognition and action. And David Rosenbaum just said, good luck, because you can't, right? If anyone knows how, please let me know. It's a really tough project. So I'm trying to make baby steps and look at where are these deficits? Is it inhibitory control? Is it an actual muscle movement generation problem? And so to study inhibitory control, as Michael mentioned, we can use a task like a go, no-go task. And so every time you see the letter X, press a button. When you see a K, don't press the button. That's the standard version of this lab task. We've got hundreds of these, if not more than hundreds. But my issue with this is that that little button press, you can generate a lot of force and a lot of muscle activity and never hit that button. And so from a motor perspective, I'm anti-button press task. I hate when I see those articles saying they study the motor system with a button task, because motor system is continuous, right? It is a continuous, not a discrete 1-0 variable. And so what I did was we adapted this into our basic 2-bar setting, and we said every time you see a colored bar, press, okay, except when it's blue. So we created our own go, no-go experiment within this force setting. They had to be rapid presentation to generate that prepotency to respond. 25% of the trials were no-go trials, and we had 2 conditions here, where the force you needed to generate was low relative to your maximum, or high. And so in adding that variation, we can also look at response planning, and do you set your parameters correctly to the target. I'm not going to talk a lot about that today, but I am going to talk about the response to this task. We had 51 adults with ADHD and 51 adults without ADHD. Everyone went through a diagnostic interview in our laboratory in order to determine their status. And anyone who took stimulant meds was off meds for 24 hours when they came to the lab. And I failed to mention, we had a button press task as well. The stimuli were exactly the same, okay? And what we found was just what you would expect. Adults with ADHD, that's the blue bar, made more errors, commission errors. They pressed when they shouldn't in the button press task. That's not news. We've got tons of those studies, okay? But what we were able to show is that in the force task, here's our low and high amplitude conditions, and the controls are in red, and the ADHDs are in blue. What you can see here in the go task is that these guys are the same. So I'm demonstrating that ADHD, adults and individuals with ADHD don't have trouble producing force. They did the go task just fine. And in fact, they scaled the response appropriately, right? So they were able to plan. But when it comes to the no-go task, here you can see that there are differences. And so this is an actual continuous variable. How hard did they press? And so adults with ADHD pressed harder when they shouldn't have pressed at all. And so could this then be an indirect measure of inhibitory control? The harder you press, the poorer your inhibitory control. That's the analogy I'm trying to make. Or the more activity you have in the motor system. We don't have physiological data on this task yet, so I can't complement that, but that's the argument that I'm trying to make here. The last figure here on the right is the standard deviation, and as you would expect, there was a lot more variability in those no-go trials as well. This is pretty characteristic of any clinical population, but ADHD in particular. My grad student is spending some time looking at this to see whether or not adults with ADHD, this variability was given by 2 or 3 trials that were really high, or whether it's a consistent kind of pattern that stays around a mean for each individual. So we're looking into that data right now. So the key thing is, how could we use this task? Why do we care, right? So we did hierarchical regression, and first thing we did was put the standard reaction time task in, and then the forced task. And then we flipped it and went the other way around. So the standard reaction time task did not predict any unique variance over and above the forced task. But the forced task contributed for unique variance over and above what was predicted by the reaction time task. So the addition of this forced measure was accounting for more variability in the sample. And I should say the predictor here, we looked at the number of symptoms from the diagnostic interview that we had. We also looked at other variables that were, related to self-report of ADHD-related symptoms and found a similar trend. So on the line of so what? It's difficult to diagnose individuals, especially when you're relying on self-report, especially in the case of ADHD. I was warned going in, you're going to find that a lot of them over-report and a lot of them under-report, and it's not very reliable, and you need 3 different observers. So if we had a biomarker, if we had something that would help us corroborate the story that we're getting, this would be really useful, right? And so in terms of translational relevance, one of the problems here that we've all been talking about today is, can we get a biomarker? Is there something that we can use, physiological data that we can use that will help us? It might not be 1 or 0, but it might help us push our diagnosis in one direction or another. If we had that thing, whatever that was, maybe it would also give us an idea about prognosis. So if you fall on this continuum with this biomarker, okay, then you have a pretty good chance of success if we apply treatment A versus treatment B, right? Which gets to that treatment response component. And so this is obviously the goal that many of us, if not all of us in the room, are really working towards right now, is finding that physiological signal that's meaningful for diagnosis, prognosis, and differentiating our non-responders from our responders. So the solution is to study a system that's well understood. That's the motor system. We understand it very well. That's not to say that there isn't work to be done, because there's plenty of work left to do. But in terms of these simple one-dimensional force movements, we understand the visual motor network. We understand what a healthy young adult, what the response should look like, and what brain networks are involved. And so then we can go ahead and do these studies without having to do the fMRI study for, right at the outset, because we can take the existing literature and build hypotheses from there. And then this might help us understand the neurobiology of the disorder and then get us back again to this idea for finding novel avenues for treatment. In terms of the applicability, we've also talked about these non-responders today, and I'm going to suggest that movement might be another way to treat mental health disorders. Not all of them, but that if we contribute or we start to understand that movement is a physiological disorder and not a behavioral problem. So my example is, you might have that niece or nephew or child of your own that you just say, oh, just sit still. And you think, if only they had the cognitive control, right? Or the behavioral control to sit still. But if it's a physiological problem, asking them repeatedly to sit still isn't going to solve that for them, okay? So maybe we just let them move around. That's been proposed by having these classrooms where the kids are all sitting on chairs, or balls for chairs. If you've ever tried this for an hour or so, what you'll find is that you're just kind of moving around in your own little space, right? It allows you to fidget. I found a lot of anecdotal evidence, but not a lot of scientific evidence for this. But this is one of the ideas. Let them move. And not just the one kid with the behavioral problem, but let them all have these exercise balls and let them move around. Can we use physical therapy? That's a novel idea, and I doubt insurance would be happy about it. But we know that the basal ganglia is involved in generating this precision grip force behavior that I was talking about. And we know that it's involved in many, many mental health disorders. And I'm sure that these neurons are not the same neurons, but it's possible. We have a lot of systems in the brain that are used in multiple functions, and so if we can target that system through movement, maybe, and I am generalizing here, maybe that will improve their behavior in other contexts. And so what I'm showing here are just some fine motor skill tasks, clothespin, picking up little pom-poms, little tasks like this that you can do to improve manual dexterity, control, attention, and whether or not they transfer, I have yet to find out. So as many people have also said today, I have a lifetime of work ahead of me, and my goal right now is to really nail down this physiological marker so that I can continue with some of these type of intervention studies looking at how movement may affect some of these cognitive measures. All right, I've got just 1 minute left. So barriers to success. Patient recruitment, especially in this area, is very, very difficult. Self-identification of mental health disorders is very difficult. So it's a difficult population to work with, even though I find it very rewarding. One thing that I'm really working towards is getting mobile data collection. So what I've got in the lab is not something I can put it on a cart, but I can't put it on a cart and get it over to the Park Avenue Clinic without a lot of logistics, right? And so having some applied scientists who can help develop novel tools that are portable, that are stupid-proof to a certain extent, so that you can have someone in the clinic who's an RA that doesn't need a lot of training on your task, that's a big deal. That will let us get patient data without them having to come to us, if we can grab it while they're already there for their treatment. And then the last barrier is recognition of team science and interdisciplinary science. And genuine teams, as we know, can really yield great findings, but they can be terribly time-consuming. They cross the boundaries of each individual's field, so their publication record may look unfocused because they've got papers that are kind of in multiple fields on a general topic. And our system, in terms of the academic system, is geared towards reinforcing work in a very narrow field, having a body of work that's in a very, very narrow field specific to whatever your field of study is, and doesn't recognize this interdisciplinary team science work. But I think we're moving forward. And then the last point I wanted to make, and I'm especially aware of after training a graduate student, is it's tough to sometimes convince graduate students that they should be interdisciplinary. That means you have to master multiple bodies of literature. You might have to take more classes. It's more work. And as a grad student, by the 4th year, you're not really interested in more work, right? And so I think we have to think about how do we teach team science to the next batch of students that are coming up? How do we make it not an extra thing for them, but how do we kind of integrate it into their training so that they see it as the right way to go about solving some of these big problems we've discussed today? And with that, I just need to acknowledge the folks who contributed to this work today, as well as some of my collaborators. Thank you.
So questions for our speakers?
Thanks for your remarks. So a couple of you spoke about the relationship between brain and behavior, and when we're talking about even some of the behaviors that we measure in the lab, go, no-go, or other types of behavioral phenotyping, can you comment about how well you think these actually translate to real-world types of behaviors that we might see in different types of conditions?
I think it depends on the task and what you want to generalize it to. So what I can say with my data is that, yes, we found that difference in the button press version, but in the force version, which I would argue is more ecologically valid, we found it's much more subtle than we thought it was. And if we use a task that's going to be more precise or continuous in nature, we might catch more of these differences between groups that then relate to that specific behavior. But the more you can adapt the lab task to that behavior that you want to, whether it's by changing the visual stimuli or the mode of response, I think the better it is to generalize that behavior.
Just to piggyback on that a little bit, I think I remember my graduate mentor saying, if you can avoid designing a scale, you should avoid designing a scale. And I think sometimes the same is true of tasks. That said, at least in, for example, social phenotyping. Like if we're talking about interpersonal behaviors, like one of the things that we've been working on recently is social dominance and social dominance representation in narcissism, for example. You have to really think about what's a phenotype that we can measure in the lab. So like we actually have people play a competition game with others where they have to be faster than the other person and they get negative feedback if they're slower. So there's always this kind of tension between like how much work do you sink into having your task be as close to the real world as possible versus what's feasible in the scanner, which I'm sure you know those kinds of problems very well. So I think for us, we try and think about what's the atomic unit of behavior that's still as close to the phenotype. So for example, like social rejection is really important in borderline personality. How do we tap into that experimentally? But it's just a delicate dance to try and find a task that is controlled enough that everybody gets the kind of same reinforcement history, but is close enough to the real world that you think it has that translational ability.
Other questions?
Dr. Neely, in terms of what you've been talking about, I don't know this field very well. Have people looked at, in terms of making that differentiation, a combination of neuroimaging along with the kinds of tasks that you have, for instance, and in other fields, often the neuroimaging result will turn up before the actual behavioral measure will tell you anything. That's anywhere where in your trajectory.
I think it would be beneficial for us. It's definitely happening in the movement disorders research, where they're looking for Parkinson's disease is a great example, right? Those dopaminergic neurons have died way before the motor symptoms emerge. And if you could move back to figure out when that happened, that would be great. So I know the research in that field is really looking at, okay, what can we get out of a structural MRI? Because that's easier for a clinician to do than fMRI. There's a lot of computation involved to get that fMRI result. So I think the push is in more understanding how do we use the structural MRI results with the clinical picture with some behavioral tests that are fairly easy to do in a clinic or other kind of situations.
Other questions? I have one. I was thinking, Dara, for your presentation, you showed some of the findings looking at the effects of puberty, and the early and late puberty sort of is operating differently for boys and girls. I was wondering if you had any thoughts on what might be going on socially for those boys and girls that might potentially account for some of the depression differences.
So most of the work on early puberty has focused on girls. So we don't really know a lot about boys and puberty and risk for depression. But the little that's out there seems to suggest really inconsistent findings. But there are some studies that support the nature of the findings that emerge from the MTA study, that there's something more protective about maturing earlier for a boy. You know, you get big and strong sooner, and that has social implications for how you're accepted and perhaps how well you do in sports, and then you're popular and so on. You're attractive and people want to date you and so on. But for girls, especially being a girl with ADHD, we don't know a lot, but what's emerging is that the ramifications or the social ramifications of being a girl with ADHD are potentially greater than they are for a boy, because there are all these theoretical hypotheses. So for example, the gender paradox, where ADHD is gender atypical for a girl. So you know, in society, we're not so, you know, we know boys will be boys and they'll be hyperactive, and that's kind of how people are used to that. But finding a place for girls who manifest some of that behavior, it can be harder. And we see in the peer literature and the, you know, the mood literature that girls with ADHD compared to boys are more rejected or less likely to be accepted by their peers, and that social dysfunction is more clearly related to these negative long-term outcomes like depression, eating disorders, suicide attempts in girls with ADHD rather than boys with ADHD. that answer your question? Yeah.
So you were bringing up eating disorders. You reminded me of the co-occurrence of obesity and ADHD, and I was wondering if you looked at whether or not body weight, BMI explains some of the depression related in girls versus boys?
In the MTA sample, it doesn't seem to come out at the pre-adolescent age. We're continuing to collect that now and are collecting the eating disorder inventory on all the girls who come in. And it seems like once in a blue moon we'll get some variability there. Might just be a factor of the age. But yeah, we'll see.